about briefly, if you can hear me there, Neph, we talked about chromosome 2, and I noticed you were really giving Drag there a run for his money. I mean, the guy was falling all over himself. I can't remember if it was Crispr that came to the rescue as usual, or usually it's either Crispr or Sam that comes to the rescue if these guys are mutable and Drag. But human chromosome 2, right? They're saying that it arose through a fusion of two ape-like chromosomes. But we know that this, apparent fusion site is actually a functional DNA element in a human gene. Did you want to expand on that? One for Guzman. I know Guzman was exposed to that through some biblical compromiser named Robert from Sentinel Apologetics. The guy won't debate it.
Sure. Yeah, I'll be happy to. Let me take a bathroom break. I'll be going about 1.5 minutes, and then I'll be happy to expound upon that. The argument that was made by the evolutionists in the hangout the other day were just their ideas from their head, things that they need to believe, but not actually scientific. And I'd love to talk about that for just a minute and explain why the real science gets denied by evolutionists because it doesn't comport with their evolution theory. Give me about 60 seconds.
Well, because if we actually look, and while he's gone, I'll fill in, Guzman. If we actually, look at recent reanalysis of the human chromosome 2, we know that it's now contradicted this evolutionary prediction, right? It kind of falls all in line with the junk DNA paradigm, and this junk DNA paradigm has certainly been overturned to the point that there's actually no significant evidence that this actual fusion exists. And what the facts are is that this alleged site, say where this fusion supposedly took place, actually represents a highly organized and functional gene. So if you actually start from the assumption of, say, human to ape common ancestry, it's evident now that evolutionists have actually made a failed prediction about the structure and function of DNA within our cells. So at the end of the day, a lot of their, strongest arguments, say for, well, in this case, ape to human common ancestry, it really has been overturned. And they'll fight it tooth and nails. They'll never give it up. There's too much at stake, as Superbad was indicating. I'm not sure if you heard any of that, Neph. I know you were.
No, I just came back about 15 seconds ago. So.
I was just talking about briefly how the human chromosome 2 is really just a failed evolutionary prediction because of the highly organized functional gene.
Yeah.
In the alleged site, right? Did you want to expand on that a little bit?
Yes. Yes, it's been discovered that there are 1,996 functional associations with biological entities spanning 7 categories that include molecular profile, organismal, chemical, functional term, phrase or reference, cell line, cell type or tissue. That cell type means cell specialization. Cells are given genetic information when they split sometimes that determines what kind of cell it's going to be. Is it going to be a pancreas cell? Is it going to be a heart muscle cell? And this is how an organism develops in their fetal development stage. As an organism develops, cells are specialized, and information in a given cell, when a cell splits, cells aren't produced just de novo in an organism. They're not just created. All cells in organisms come into being because a previous cell splits and doubles it splits the genome, and the cell splits into 2 cells, and each one gets a copy of the genetic material. That's how new cells form, not by a new cell being created, but by a cell splitting into 2. So that's how cells are made. That's called, and When that happens during fetal development, like during, when your mother has you in your womb and you're developing over a period of 9 months, different cells specialize to become this kind of tissue, that kind. There's over 250 some odd tissues types in a human being. So a cell is given a specific genetic information twist during the cell division that tells that you're going to become this kind of cell. Instead of a brain cell, you're going to be an intestinal wall lining cell, or you're going to be a bladder cell, or you're going to be a nerve cell, or an artery cell, or a heart valve cell. That's called cell specialization. So these 1996 known functional associations of the genes that I'm about to explain include cell specialization, genes, proteins or mitochondrial RNA, I'm sorry, and they relate to 20 different data sets. Now, here's the thing. This imagined chromosome fusion site, evolutionists say, well, human beings and chimpanzees have during the evolution of mankind, the chromosomes fused, and that's why we find this profound evidence for evolution, because the difference in chromosomes between humans and chimps is explained by 2 chromosomes having fused in the history of man. Okay. Now, the problem is that science has discovered that, imagine in your mind, from the left to the right, we have a line going across your screen, okay? And you draw a line down the middle, a dot in the center of that line. And let's pretend that's the fusion site, okay? Now, to the left of it is the human chromosome. To the right is another human chromosome, and they fuse together, right? So now, in the dot in the center is where they fused. And that's what evolutionists claim is evidence that humans and chimps are related. But here's the major problem with that. There are genes that span all the way across that dot in the center. Gene sets, not just a gene, numerous genes that cross over that fusion site, that dot in the middle. And they have all these different types of functions, 7 categories: molecular profile, organism, chemical, functional term, phrase or in reference, cell line, cell tissue, protein, mitochondrial RNAs. All these types of the information in the genes that span across that dot, that fusion site, supposed fusion site, relate to these different categories of critical function in a human being. Now, it gets really deep. Kind of try to stay with what I'm about to say. I want people who know nothing about what I'm saying to understand, because there are people who say, man, I don't have a clue what you're talking about. And it's not that I'm so smart or that I'm telling you stuff that's really complex. It's just that once people get engrossed in this kind of thing, they tend to use terms that some people who have never encountered any debate about evolution or creation, they don't know what they mean. So they don't know what the heck you're talking about. I want to make it simple so everybody can understand the failure of the evolutionist claim that this is a fusion site. Okay. So the dot in the center is where these 2 chromosomes, these 2 giant bundles of genetic information, came together and merged, and they became fixed and stuck to each other, and they're permanent. Now they're forevermore together. Okay. But the problem with that is that there are many complex genes that read, if you read them left to right, like you're going to read a sentence in a book, and you start to the left and the words go on to all the way to the right until you find the period all the way on the right side, right? The problem is that sentence goes across the dot, the supposed site where the 2 chromosomes fused. You see what I mean? Who wrote the sentence that goes all the way across the spot where there was supposedly two entirely different and related books that were snapped together. How's that possible? How do you have a coherent sentence? Not only does the sentence, is it a coherent one, but it relates to functions that are critical for the book on the left and the book on the right to even exist. Else, they couldn't function. And some of these functions are, one of them is that these codes, these genes, and this set of genes is called DDX11L2. And it produces, one of the things that it produces is what's called helicase. Now, helicase is a really complex set of molecular machinery stuck together that produces a single machine. And that machine duplicates the DNA molecule in your cells. In other words, the DNA, this machine, it's really complex, made of proteins and numerous components, latches onto the DNA and it starts to duplicate it, to make a copy, because you have to have the duplication of the DNA molecule going on in the cells or you don't you can't survive. The DNA has to be duplicated. And it's duplicated constantly. In your body right at this moment, there are probably hundreds of thousands of DNA molecules that are being duplicated by this quote-unquote helicase machinery at this second. It's constant. Okay, so this machine is, let's imagine an automobile engine. Okay, so let's imagine that one component in that automobile engine is its heads. That is the block of the heads with the valve train in it, in an overhead cam valve engine. You've got the camshaft, you've got the valve springs, you got the rockers, you've got the valve springs, you got the valve guides, you got the valve stems themselves. All that is one component of the engine, and the other is a block, and then another component is the crankshaft. And then another component, let's imagine that if we could liken all the piston rods with the pistons and piston rings connected, is a separate component. All the pistons and the churning component inside the engine. Okay, that's another unit. Okay, that's kind of what we're talking about when we're talking about helicase. It's numerous complex molecules arranged in a specific manner so that each one has functions, and then they are different ones are put together to comprise a much bigger component, like the heads and the block and the crankshaft with the piston rings and everything, connecting rods and pistons and piston rings are all a separate unit, put together make an engine in your car. So these helicase machines combined together create the helicase machine, and it duplicates the DNA molecule. It's critical to the function of your life. If it didn't exist, you would not exist. And it spans the dot in the middle of the page. In other words, it starts on the left and it ends on the right. It goes across the dot. Now, if the dot represents the spot where one chromosome on the left and one chromosome on the right came together and fused, these genes with all these different functions, and one of them being that it produces this macro collective, outrageously complex collection of molecular machines that could together produce a machine called helicase that duplicates the DNA molecule, then it's not plausible that part of the DNA, the helicase, or any of these other functions like cell specialization, that information on one side, information on another, happen to block together, and then boom, we get the processes. That's not plausible. That's out. Can't believe that. Therefore, if this was actually a fusion site, here's what would have had to have happened. The genes that create the helicase and the other functions that are important for cell specialization and whatnot had to have been made so that they span across that fusion site after the fusion took place. And that would have had to have happened by random genetic mutation. And in our last hangout, I asked Crispr and a couple other people, what's the reason for believing that? And Dragnock Silvas simply refused to answer my question. Their response to me was, well, Nephi, when you understand enough about science for me to talk to you, give me a call. That's the kind of response that we got out of them, instead of an actual scientific response.
And then meanwhile, we asked Drag for hours to come on and have a simple discussion with you. I mean, at least we'll give CRISPR credit. You know, he certainly talked with you for, you know, extended amount of time. But that Drag, he must have Neph nightmares.
Well, I mean, instead of answering the question or talking about it intellectually, we get these ridiculous hand-waving, you know, well, I can't respond. Obviously, they can't respond against the real science with their philosophy. So the way to fix the problem for them is to prevent, because they can't answer, they got to have a tool to explain why they can't answer. Like, Nephi, when you understand cell mutation, let me know. This is their way of dealing with it. They can't deal with it, so they come up with these childish responses. So here's the thing, that these genes span the supposed fusion site. They have critical functions for the existence of the organism. CRISPR actually told me they're non-functional. That was his way of responding to the evidence, that they're non-functional. The problem is that these functions are listed in a genetic database as functional. So what he's doing there is saying, science isn't true. What I believe and what I tell you is true. I don't care that they're documented as having function and they're recorded in genetic databases as being functional. I'm telling you, they're not functional. So what we've got is the belief system of the evolutionist turned into supposed science, which refutes actual, observable, tested, repeated, documented science.
Well, it's just like, you know, the apparent pseudo gene that evolutionists like to harp on or to promote. You know, supposedly this remain of a gene devoted to egg yolk production that's apparently in our DNA in exactly the same place that evolution would predict. But just like many other, you know, specific genetic markers, specific pseudogenes, chromosome 2, we know that this recent research has actually exposed this as nearly impossible to reconcile with the actual facts.
Completely
impossible. Fight it, you know. Yeah. But at the end of the day, this supposed evidence, say for the egg yolk gene, it's so pathetic, it's so sad that it's hard to imagine that anyone could have actually entertained this, you know, hypothesis.
Yeah, they shouldn't, because the germ lines that produce different features in one organism and another don't line up with that egg germline cell thing. That idea was discredited. In fact, over 100 years ago, even almost before they even understood what DNA really did, that idea was already refuted. Well, let me finish about what I was saying briefly, if you don't mind. The supposed fusion site is spanned by critical genes that produce machinery that's critical to the function of the organism, and cell specialization information overlapping in a nested information that does wonderful 7 different categories of critically important information in the human cell and organism. Okay, but now, as I pointed out, those overlapping and nested genes that span the supposed fusion site would have had to have been produced by evolution, by mutation and natural selection after the fusion, because they're there, right? So, but the problem for that is that science doesn't document any reason for believing that random genetic mutations can build genetic information, that it can create overlapping and nested information, which requires the sharing of base pairs between one sequence of information, quote unquote gene, and another sequence of information, quote unquote gene. We have no scientific reason to believe that malarkey at all. All we observe is mutations destroy. But evolutionists, in their wild, crazy, mythological, religiously ardent, and fervent belief system, believe that mutations can do this kind of ridiculous thing. That mutations are going to design, and mutation would have had to have designed all those overlapping and nested genes in that DD11 XL site in order for that to have happened after the supposed fusion of these two chromosomes. But there is nothing in science, not one thing, that makes anybody believe there is a scientific basis in believing that random genetic mutation can, one, create information, two, it can create overlapping and nested information, whereas information that in one sequence shares base pairs with another sequence of information that spans even a greater length, perhaps, than it is, wider to the left and the right.
There real quick, and it seems like, because in the chat, Crispr, it seems like if you put it all in a nutshell, he's just saying, and I imagine many other evolutionary scientists, well, there's no actual evidence for function.
Yeah, but it's already in genetic databases as functional.
Yeah.
So he's denying science right there.
Would you say a lot of them are science deniers?
All evolutionists are science denialists. When you show them science and it contradicts their philosophy, they'll deny the science to hold to their philosophy.
I think based on their arguments, whether it's spurious transcription or, there's function, but it's not a useful activity, I think they're just missing the bigger picture and the actual point, Neph, because first off, preliminary biochemical evidence for function does not exist merely, say, for, the examples of pseudogenes that we were talking about. It actually exists for at least 80% of all pseudogenes in humans. And then the other 20% may actually still yet to be found, functional in some human tissue or under some physiological condition yet to be studied. And there are many. So really, it all comes down to the fact that many non-coding RNA genes, say like these pseudogenes, are only expressed under certain conditions. And then we get into the whole embryonic, placental type functions. I mean, junk DNA really is dead, but why do they have to hold on to it so strongly?
Well, they do. But listen, here's an interesting thing too also. CRISPR said that they're not functional. Science demonstrates they are. They've already been documented in a database, a genetic database that says they are. But let's pretend for a second that his claim, although already fallacious, were true. That these genes, although they do code for helicase and other things, are not functional. Let's say they're there, but they're never used by the DNA. Let's pretend that that were true, but it's not. But that's what CRISPR said. Let's pretend that's true. That still doesn't explain the existence of them. You're still wanting us to believe that random genetic mutation, then after the fusion site, listen to the insanity of this now. Listen carefully. CRISPR is still expecting us to believe that random, unguided, undirected molecular interactions will create the genetic information that just so happens to mirror exactly the same genes that code for the helicase, the outrageously complex helicase machine for a gene that's found somewhere else in the genome exactly like it, and create that after the fusion site. And it's going to do, random mutations are going to do that after the fusion site. They're going to build the same genes that create the functional helicase machinery. I mean, what in the world could possibly make that rational? Now, the only explanation that he could come forth with after this is, and he never got to this, but this is where I would have to take him if I questioned him and pummeled him with science. You would have to come to this. Well, it's a duplicate mutation. Genetic information over here was copied and inserted and rewritten over the base pairs that span the fusion site, rewritten over it. Okay, but when that kind of thing happens in genomic information, it doesn't happen by rewriting an entire set of base pairs. It happens by copy and paste, an intelligent design process, just like you do with Microsoft Windows. You copy and paste things. That can happen. That is a real genetic process. Information can be copied by the genome using algorithms, or go way over here to the other side of the DNA in a new location and cut the DNA molecule, paste that snippet of information in, fuse it, and connect the ends again. And now you've got a new chunk of DNA in there that was copied from one place and pasted into it on the other. Nobody knows of any genetic process where information is copied on one place and then machinery connects to the DNA and overwrites all the information that's present to rewrite that as something else to create the helicase. It's not known to science. That does not happen genetically. So that's the only place somebody like CRISPR could have gone, is to say that the reason we find these specialized genes that, like, for example, the ones that code for helicase that span the fusion site, is because information was copied from one place in the genome and then it rewrote all the base pairs across the fusion site to make those genes. That is not a genetic process. That doesn't happen in DNA. Insert, copy, and paste into, move the information down some and snap a new piece in, that happens. Copy and rewrite a whole section of genetic information for new genes doesn't happen. What that would also do is completely destroy all the genetic information that it copied over. That's just not a process in the DNA. So again, the evolutionists, when you ask them these kinds of things and show them this kind of evidence, they have to continuously, with their rebuttals, with their attempt to respond, continuously back themselves into a corner, to a smaller corner, to a smaller corner, to a smaller corner, to a smaller corner, each one being scientifically false, to a point where what the ultimate answer that they have to give to explain, for example, these genes, like the one that codes for helicase, spanning the fusion site, is something that's so utterly absurd and obviously scientifically false that everything that they've said from the beginning about the subject was just their ideas in their head and not science. So the fusion site cannot be a fusion site. In summary, let me sum it up really quick. In summary, it can't be a fusion site because the place where these chromosomes supposedly join together, what spans across that fusion site, just like a bridge going across a river, is numerous genes that have 7 different categories of critically important function to an organism, that one of which is the codes for the machinery, very complex machinery that duplicates the DNA molecule. You ought to see a video of that, how that operates on the internet. It'll blow your mind. The machinery. Look for that. It'll blow your mind. That machinery is coded across this fusion site, and it's not something copied and pasted in, and it can't be something that was copied and rewritten over, because that doesn't happen, except in the immune system to make an enzyme. That's it. That's the only copy and rewrite new stuff. It's the enzymes that do that. That's the immune system. Okay, so the idea that it's a fusion site is not plausible, and the arguments put forth by people like CRISPR and others that try to explain this away can be backed down by asking one question after another to demonstrate it's not only scientifically implausible, it's utterly absurd, and science kills that idea because of one thing, and then I'm done. Sequences of information that code for special critically important functions in a human being, including the machinery that duplicates the DNA molecule, spans all the way across that supposed fusion site. And that can't have been created before or after a supposed fusion, because there's no genetic mechanism for that. It just gets that simple. It can't be true.
No, I find it funny because if you look at a lot of their, purported evidence, we now know human chromosome 2. Well, you know what? That site is actually a functional DNA element in a human gene. These pseudogenes, the fact that they say humans and chimpanzees share genetic mistakes, aka pseudogenes. We now know these pseudogenes appear to be functional DNA elements, not actually mistakes. What's another one that they use that has failed? even the fact that, they say human to chimpanzee genetic identity is what, 98 to 99% similar. But now, based on the data, genetic identity is only what, 88% similar. And to put that into perspective, that's over 400 million DNA differences that actually exist between the two species. So, I mean, how do they reconcile that all their, major evidence for Pons come to people evolution is just falling apart bit by bit?
It's